We were just waiting for a few more participants to join the webinar, but by my watch, it's now 12:02, so we will get started. So welcome, everyone. My name is Susanna Turner, and I'm the Research and Innovation Development Manager for the National Pig Centre at the University of Leeds, and I'm thrilled to be chairing this webinar today. A little bit about me and my role before we begin. So I've been at the university for just under 5 years now, and I joined around the same time that the National Pig Centre was being built. And my role is to bring together researchers and external industry partners from different disciplines to both develop and deliver research and innovation projects. Now, they broadly focus on the UK pig industry and how we can enhance its sustainability. So I work very closely with Dr. Katie McDermott, who is presenting today, alongside the academic director for the National Pig Centre, Professor Lisa Collins, who I'm sure many of you will be familiar with, and also a brilliant team of researchers and technicians who all contribute to making the centre a real powerhouse for UK pig research and innovation. It is my pleasure to welcome you to this virtual seminar on sustainable livestock and the National Pig Centre, hosted by the university's Global Food and Environment Institute and presented by Dr. Katie McDermott, who is a lecturer in sustainable livestock here at Leeds. Katie completed both her PhD, master's and undergraduate degree at Leeds before joining as a research fellow in animal nutrition and physiology in 2018, specialising in the gut microbiome of pigs. And her broad interest is in animal science and nutrition and understanding how the efficient utilisation of feed is influenced from a gut microbial perspective, alongside the ways in which the gut microbiota interacts with the host animal in terms of health and disease. Over the last 4 years, Katie's specialism has developed to focus on improving the sustainability of pig production, with a particular emphasis on the early stages of a piglet's life and finding sustainable ways to improve animal health and productivity whilst reducing feed waste, minimising land use, and decreasing environmental pollution. Her research spans all aspects of pig production, and I'm sure we'll hear more about all of these exciting projects shortly when I hand over to Katie. But before I do, I just want to take this opportunity to provide a little bit of information about the Global Food and Environment Institute, or GFEI, as we like to call it, who have kindly organised this webinar today. And this forms part of a series of spectacular talks from some of our academics here at Leeds. GFEI itself is an interdisciplinary research community that brings together members from across academia, industry, and public policy to work on the integrated challenges of food security and climate change. If you are interested in hearing more about GFEI or some of the other seminars that they'll be running as part of this series, I think one of my colleagues will be posting some links in the chat function here, so please do take a look. And we'll also be sharing information via Twitter and LinkedIn, so please do connect with us on social media if you haven't already. And then lastly, some friendly housekeeping before we begin. So please do ensure that your microphones and cameras are switched off throughout the seminar, just to avoid any interference for our presenter today. Our seminar is being recorded, and that recording will be available via our YouTube channel. The presentation will be around 20 minutes with a 10-minute Q&A session at the end. So please, can I ask that you pop any questions for Katie as we go along using the chat function below, and ensure that your question is addressed to everyone so that we can collate those, and then I'll come back to them at the end of the presentation. And now finally, without further ado, I'm going to hand over to Dr. Katie McDermott. So thank you very much, Katie.
Right, hello everybody. Thank you, Susanna, for that lovely introduction, and thank you to GFEI for asking me to speak to you today. Lovely to see so many of you here. So what am I going to talk to you about then? So the first half of the webinar today will be an overview of the capabilities of the National Pig Centre. So I'll talk you through all the different parts of the Pig Centre, the technology we have there. And in the second half, I'll give you an overview of the kinds of research we have going on at the moment, areas we're keen to explore, and then, as Susanna said, we'll finish up with a Q&A. So the National Pig Centre officially opened its doors in November 2019, and the centre was a result of an £11 million investment from the University of Leeds and CL, which is the Centre for Innovation Excellence in Livestock, with funding from Innovate UK. So the NPC, as we affectionately call it, is the UK's most advanced facility for research into pig nutrition, behaviour, welfare and health and production systems. It's home to both an indoor and an outdoor herd, both at commercial scale. So the NPC is the only UK research facility for outdoor production, and it's unique in Europe for having both indoor and outdoor pig production systems on the same site. And that's really fantastic for us as researchers. So approximately 40% of the UK's pig breeding herd is housed outdoors, and this facility allows us to make like-for-like comparisons between these two different production systems, whether that's us looking at nutrition, management, welfare, or the environmental impact of pig production. The NPC itself sits within the wider university farm, where we have lots of ongoing work on crop production, soil health and contaminants, regenerative farming, and much more. And we're always looking for ways of incorporating the pigs into this wider agricultural research work. So at the NPC, we're co-developing research in collaboration with both academic and industry partners to try and address the challenges that are facing pig production. And before I get onto the pigs, which we'll come on to in a second, I just wanted to point out another couple of things. So the National Pig Centre is also home to the Nigel Bertram Visitors Centre, which can be hired as a conference centre for meetings and events. We have CCTV throughout the pig unit, so we can use the conference centre to offer virtual tours as well as our in-person tours. So that's a really fantastic tool as well for us for teaching. And we have on-site laboratory facilities as well for any sample preparation. So onto the animals then. So indoors, we have 440 breeding females, which are our sows. We work on a 3-week batch system. So what that means is that every 3 weeks, somewhere between 50 to 60 sows will give birth to their piglets. We're averaging around 15.7 piglets born alive per litter. So that gives us around 900 piglets born on farm every 3 weeks indoors. So we're producing just over 12,000 pigs a year on average. We run the farm commercially, so that's making sure that we're relevant to our industry research partners, and we take these piglets from birth all the way through to market weight. At the same time, we have our outdoor unit. We have 220 sows here, again on a 3-week batch system, and this is in sync with our indoor herd. We currently sell these piglets at weaning, so that's about 28 days of age, and when they're weighing around 8 kilograms, and this is under Red Tractor and RSPCA assured schemes. So my plan is now to give you a quick overview of each part of the farm, and we're going to start in the sow house. So this is where our pregnant pigs are. So this is where we house our pigs through gestation. So our sows are housed in one big dynamic herd on a straw-based system. So what that means is that we have sows at different stages of pregnancy living together. And we're able to manage this quite easily through our electronic sow feeders, which I'm hoping you can just about see, if you can see my mouse down the side of the room here. So all our sows have electronic ear tags. So when the sow enters the feeder, the machine knows who she is, and it will provide her with the diet allocated to her, and it will keep providing feed for the day until she's reached her programmed amount. We can provide different diets to the different sows without having to split them into different parts of the sow house. Once she's finished eating, we can program the feeders to separate her from the rest of the herd. So for example, if the sow needs a routine vaccination or if she's due to give birth, she can enter the pens on the left-hand side of the feeders. And if she's not programmed, she will rejoin the main herd on the right-hand side. And what that means is that the sows are ready drafted out, ready for the farm staff for when they need them. We also have an automated heat detection system. So there is a boar pen around halfway down the row of feeders there. And there's essentially a hole through which the sow and the boar can touch noses. And the hole has a sensor on it, which monitors which sows are coming to see the boar using her ear tag. So if she comes for 3 or more prolonged visits in a day, the system will flag her to the farm staff for check-in, as she may not be pregnant, so that's saving the farm on any empty days. All of the sows on site are artificially inseminated, and they will stay in the sow house until about a week before they're due to farrow or give birth, at which point the system will draft that group of sows out of the herd and they'll be moved through into the farrowing house. So once our sow is in the farrowing house, this is where she will give birth and nurse her young. So our sow will stay in this building until her piglets are weaned at approximately 28 days of age. The sow will then re-enter the breeding herd, so back into the straw barn, and the piglets will move on to the nursery. So there's 6 rooms with a total of 120 individual farrowing pens. And as you can see from the photo here, our sows can either nurse freely or they can be temporarily restrained within what's called a farrowing crate. And this is just for the first few days after she's given birth to protect her piglets whilst they're at their most vulnerable. And I'll mention that in a little bit more detail later. Each sow is fed to her individual feeding curve, and this is based on her age, her weight, and we can individually feed each sow through our air feed system, the pipes of which you can see at the top there. Each pen is fitted with a CCTV camera, so we can monitor the behaviour of mum and her litter 24 hours a day, 7 days a week, day and night. The piglets have a separate, what's called a creep area, and essentially this is their heated den where they can rest safely from the sow. So you can just about see them in the bottom corners of the pen down here. So these are heated with a heat mat to keep the piglets warm, again, particularly important in those first few days of life. All our pigs are electronically ear tagged at birth, so this allows us to track them throughout their lifetime. We weigh all of our piglets as standard within 24 hours of birth, at 7 days of age, and again at weaning to monitor growth performance. And we can link that tag through to their health records as well. Once we've weaned them from mum, the piglets move through into the nursery, and they'll stay here until they weigh approximately 35 kilograms. We have 9 different nursery rooms, and each one can hold up to 240 piglets. And there's 2 different types of room to meet different research requirements. So we have precision rooms, which you can see in the photos here, and we have rooms with flexible penning as well. So the precision rooms are fitted with technology that allow us to record individual feed intake, the amount of time each pig spends at the feeder, water intake, and body weight for each individual pig within a pen. Down at the bottom, you can see the 2 pigs having a drink, and at the same time, both pigs there are being weighed. So this is generating thousands of data points for us each day. Feed to these pens, again, is programmed and delivered via the air feed system, so coming through those orange funnels at the top. And pigs in these pens are in groups of 20. The flexi penning rooms allow us to house pigs in different size groups, so we can have them in 5s, in 10s, in 20s, and this is to be flexible to meet different research requirements. And in those particular pens, we can bag feed the pigs in the first couple of weeks, or we can feed them through the air feed system, depending on different trial diets and the volumes of feed required for each individual trial. Each pen also has 2 different water lines, so we can do water treatment trials as well. And as with the sows, there is CCTV over these pens that will allow us to do behavioural analysis. So once our piggies have reached 35 kilos, they move on through to our finishing buildings, and they'll stay here until they reach market weight and are sent through to commercial abattoir. Again, different types of room. We have the flexi penning and the precision rooms. So these precision rooms house the pigs in groups of 10, and this time the pig is weighed each time it eats. So we'll have the weight of the pig and the feed intake for each individual animal within the pen. Again, 2 separate water lines for water trials, and one of our rooms has individual slurry pits under the pens, which allow us to have a look at the effect of diets on nutrient excretion as well. We have good relationships with the abattoirs that we use, so we can follow those pigs through to collect slaughter data and analysis and meat quality as well. And last but not least, we have our outdoor pigs. So as I mentioned earlier, this is the only outdoor research facility for pigs in the UK, and it's the only research centre in Europe to have both indoor and outdoor on the same site. So on our outdoor unit, we have our gestating sows, so our pregnant pigs, and we also have the sows that are nursing their litter. Outdoors is slightly different. We house the sows in groups of around 30 animals each. So we've got about 7 groups, and each group is at a different stage of pregnancy. They share a field with what's called an ark. So that's the metal house you can just about see behind that pile of straw there. And just before the sows give birth, we then move them into an individual farrowing paddock where they will stay until their piglets are weaned. So that's 4 weeks of age. Each individual paddock has its own ark with a straw bed, and they each have a feeding stall in as well. And the benefit of the feeding stall is it allows us to feed mum, close the gate behind her, safely enter the pen to tag and weigh piglets whilst mum's having her breakfast, and then we can let her back out again. Also have CCTV capability in the ark, so we can record farrowing behaviour as well. We can individually feed sows through both gestation and lactation for research trials. So whistle-stop tour of the facilities there. The picture here is showing you those individual arks as well, a bit better than the one with the gestating sows. So for the sort of second half of this webinar, I just want to go through with you some of the research activities that we have ongoing at the National Pig Centre around sustainable livestock production. And I've grouped these into research interest into a few different categories, so I'll give you an overview of those now. So first, we have feeding and sustainable alternatives. So for us to have a sustainable system, it's really important that the animals are using that feed efficiently to grow, and that the feed we're giving them is as sustainable as possible. An animal that's efficiently converting feed into meat will take a shorter amount of time to reach market weight and will excrete less nutrients, potential pollutants, so your nitrogens, your phosphoruses, into the environment. That animal will also use its feed more efficiently, so it means that it lowers the cost of production because it's using less feed to reach the same weight. So this is ensuring that our system is going to be economically sustainable whilst we're minimising environmental impact. So we work across a wide range of topics under this heading, and we're always keen to work with others to target these questions along with others. So a big area for the pig industry moving forward will be looking at the protein source of the pig feed. At the moment, we rely on soya, which is imported from South America, and there's concerns over the sustainability, both from the air miles that the soya travels, as well as the growing of the soya itself, where it's involving land use change. There's a lot of interest around the use of things like homegrown crops. So our legumes, our peas and beans, which could be used to substitute some of the soya in the diet. And there's lots of scope here for research on quality of alternative proteins, the amino acid profile of them, how they affect the growth of the animal, and also how they affect the gut health of the animal as well, which is particularly important in the newly weaned pig, which is quite sensitive. We do a lot of work with industry partners, particularly looking at alternatives to zinc oxide, and this is in the diet of the newly weaned pig. So for those of you that aren't aware, zinc oxide was permitted at therapeutic levels, so quite high levels in the diet of the pig for around 2 weeks after it's left mum. And this is to promote growth and to improve the health status of the animal, so essentially reducing diarrhea risk. And it reduces our reliance upon antibiotics as well to treat sick animals. However, zinc was banned in both the UK and the EU from June last year, as it can build up in the environment or it can have some toxic effects, and it also promotes some antimicrobial resistance risks as well. So there's a lot of work going on to see what the additives we could use to try and help the young pig through this really stressful weaning transition. And one area we're particularly interested in is the use of bioactive rich food products, so keeping this circularity going. We're also interested in understanding things like individual animal variation. So what causes differences in efficiency and environmental impact, and how can we think about trying to manage it? And tying in nicely with that is individual animal feeding requirements. And this is an area that my colleague, Dr. Amy Taylor, is particularly interested in. So should we be meeting the individual nutrient requirement of the individual animal, as opposed to the average animal for that stage? And if so, how do we go about doing it? So this will mean that we make sure that we're not over or under supplying nutrients to the animal, which will cause inefficiencies in growth performance, reduce gut health, and affect the animal's environmental impact as well. And with the precision feeding technology that we have at the NPC, we really have the capability to dig down into this. Health and welfare. So you can't have a sustainable system if you don't have healthy animals. And an animal with poor health and welfare won't grow as efficiently. It will require more antibiotic usage throughout its lifetime. So it's really important that we're making sure that our animals are as healthy as they can be, and that we're meeting all of the requirements to maintain good welfare for them as well. So one of the projects that I'm working on at the moment is looking at temporary crating. So when I showed you the farrowing house, I said that we can allow mum to move freely within the pen, or we can close her into a crate to protect her young. Most of the indoor pigs in the UK will be in a farrowing crate whilst they nurse their offspring. And there's a really big push from consumers to ban the crates and cages from animal production. And this also includes the farrowing crate. If we get rid of the crate completely, we'll improve mum's welfare, we'll allow her freedom of movement, the opportunity to express some of those natural behaviours, but it's also likely to reduce the welfare of the piglets who are more at risk of crushing mortality. So the project that I'm working on is looking at how we can temporarily crate mum just for those first few days of the piglet's life to try and minimise the risk to the piglet, but maximising mum's welfare. So after those first couple of days, she'll be released from the crate, giving her free movement in the pen for the rest of that 4-week period. Susanna mentioned to you, my background is largely around the gut microbiome. And this is something that I'm always keen to continue developing. Particular interest is in the young animal, particularly the piglet. So how can we manipulate all those bugs that live in the gut to maximise health, to maximise growth performance, whether that's through the diet that we're feeding to those animals, through the interaction between piglet and mum, or the environment that the piglet is reared in. We do microbiome work across all life stages, but my particular interest is that really, really early life when the gut bugs are at their most manipulative, habitable, whatever the word would be for that, adaptable. We've just received some funding from BBSRC in collaboration with the NC3Rs to develop an in vitro model of the pig hindgut, which is called RoboHog, which we'll use to complement the animal trials at the NPC. And that will allow us to look at how an additive, a feed ingredient, a pharmaceutical will affect the microbes of the hindgut without invasive sampling of the animal or relying on fecal samples alone. So there's lots and lots I could talk to you about the work we're doing here, but in the interest of time, I'll try and keep it brief. So as I said, I'm interested in early life. I have a PhD student working with me at the minute, looking to understand the variation between sows and their litters. So what makes one particular litter perform well compared to a litter that performs not so well when all those other conditions are controlled for. So we're trying to improve the health, the growth efficiency of those piglets as early in life as possible, tying back into that sustainable growth that I mentioned on the last slide. We have a lot of interest within the group in understanding the cost benefit of indoor versus outdoor production. So as I say, UK is unique, 40% of its breeding herd is outside, and we're uniquely placed at the National Pig Centre to explore those interactions between performance and environmental conditions. And along those lines, we have expertise and interest in heat stress, which is becoming more and more important to UK production each year with the warming climate. We also have expertise looking at the use of technology. So how can we use cameras and precision feeding technology to improve our production system? For example, we've had some really exciting projects looking at whether we can use technology to predict ill health before we actually see clinical signs in the animals. And last but not least, we have members of the team that are looking at the environmental impact of pig production. For example, we're looking at ways in which we can improve the quality, the usability of the waste that the pigs produce in a safe way. We have an ongoing project at the moment, part of which is looking at how we can incorporate slurry into a circular economy, with a particular focus on the risks and the mitigations for the spread of antimicrobial resistance genes. We also have an N2 applied unit on the site, which is in this brown storage container here, which is able to alter the profile of slurry to minimise the emissions in the form of ammonia and methane, I'll admit I'm not an expert on this bit of kit, so if you have any questions on the end to applied, please let me know and I can pass you on to the right person to talk to there. But we also have projects across the wider university academic team, an interest in looking at how we can most effectively incorporate our pigs into things like our crop rotations on the university farm, looking at their impact on soil structure and soil quality, as well as examining their roles in things like regenerative agriculture. But environmental impact is always on our minds when we're working with these animals, and it's really important to make sure that we're promoting environmental sustainability as well as economic sustainability. So without further ado, if you would like to contact us, if you would like to find out more about the work we're doing or to inquire about working with the MPC, the email address to contact us is there, or you can find us and give us a follow on Twitter as well. And with that, I'm happy to take any questions.
Thank you very much, Katie. That was a wonderful presentation, and I'm sure there'll be many people on the call today who may or may not even be familiar that we have a National Pig Centre, let alone what we do there. So it's really great to showcase the research that we're doing, and I'm sure we'll have many questions in the chat bar. Thank you also for keeping perfectly to time. You're making my job very easy. So we do now have around 10 minutes to take some questions from our audience members. Thank you to those of you who have already popped a question in the chat bar below. Please do keep them coming in as we go into the Q&A session. But in the interest of time, I'm going to go straight to the chat bar now, and we'll try to get through as many of these as possible. So I'm going to start from the top and work my way down. So to kick things off, the first question is there any way to monitor the well-being of pigs through biological markers? And if so, is there a difference between the outdoor and indoor herds?
Oh, fab question. So one of the things I'm actually working on at the minute is looking to monitor cortisol through fecal samples. So this is the stress hormone. So is there a way of monitoring biological markers? Yes, hopefully. The cortisol seems to be picking up really well through fecal samples. One of the reasons that we're looking at doing that is because cortisol traditionally would be taken from a blood sample, which obviously is invasive, or saliva samples. With saliva, one of the problems is the pig gets quite excited when you go into the pen with something for it to chew, and that can increase the amount of cortisol that you see in the saliva at the same time. So monitoring through fecal samples means that we avoid causing any peaks in cortisol due to human presence. I haven't looked between indoor and outdoor specifically, but there's definitely scope to progress through with that. Really interesting to follow up on.
Thank you very much. I'll move swiftly on to the next question, which is around outdoor pigs. So does the term refer only to how breeding sows are managed? What happens to their piglets when they are weaned? That's a good question. And are they ever kept in nurseries and then grown and finished outdoors? So is pork ever marketed as outdoor reared? I think that can all be covered probably in one swift answer, can't it?
Yep. So the 40% is the breeding herd. So 40% of the breeding herd is outside. Our piglets, when they're weaned, are sold on, which I believe is to indoor production. Majority of your growing and finishing pigs in the UK will be indoor grown. And then there's always good questions to ask around the marketing. So there's outdoor, I can never get the right word, there's outdoor born and there's outdoor reared, and they both have different amounts of times that pig has to be outside for, but it doesn't necessarily mean that pig has been outside for its entire life. You can grow and finish outdoors, and we would like to get some tents to do that ourselves down the line. So it is in the pipeline for us at the MPC, but the majority of the pigs will be grown and finished inside, and that's all to do with the growth efficiencies. So
the question around whether they're kept in nurseries and then moved outdoors, it's probably more the other way round, if that's my understanding is correct. And then with regards to pork ever being marketed as outdoor reared, do we see that on our supermarket shelves?
I can't remember what the legislation is off the top of my head. There's outdoor born and there's outdoor reared. One of them means until they're weaned, they're outside. One of them means that they're outside a little bit longer. But if I remember correctly, so don't quote me on it because it might be wrong, but if I remember correctly, it doesn't necessarily mean their entire life was outside, just that a certain amount of weeks were.
I think we'll all be paying closer attention to the pork that we buy on our supermarket shelves now. Thank you, Katie. Moving swiftly on to the next question. Do pigs and piglets get stomach ulcers? So this particular person looks like they've had a background in horses and foals, who obviously do get stomach ulcers. And if so, what causes them and how can they be managed?
Yeah, good question. There is ways of looking at ulceration in pigs. From my understanding it's not as big of an issue as it is, as you see in horses. Sometimes different types of feed, bacteria in the stomach can cause them. You tend to see them, if you do see them, at the top of the stomach where it meets the esophagus. So yes, they can, but nowhere near as big as an issue as it is in horses. Whether that's different ways of managing stress between the two, I couldn't tell you, but yeah, so yes, they can, but to not as much of an extent as you would see in a horse.
Very good, thank you. The next question is an interesting one. Do you ever have open days where people can look around? I think we can both answer that, but I'll let you go first, Katie.
So yeah, with the pig unit being biosecure, we don't tend to have, say, the general public, for example, coming around for a tour. People from the university or industry can arrange to come on tours of the unit. We can also do the virtual tours as well, so you can come out to the centre and have a look on the CCTV cameras. But open days as such, no. But Susanna, I'll let you pick up on that.
Yeah, to be honest, I think we're kind of open to having discussions around what we can and can't do. In the future, we'll certainly be hosting events at the National Pig Centre, where we'll be inviting as many people as possible across the industry, internally and externally. But it will all be obviously contained within a very safe environment to protect our pig herd. It's easier to show the outdoor pigs than the indoor pigs, simply because of biosecurity, and we have a limited number of showers. And for anyone that's been in a pig unit, you'll know that you have to fully shower in and out, mainly to get rid of the smell at the end. So we have some sort of limitations there around what we can do. Hence, we've developed a virtual tour of the indoor centre, which we can run from our seminar room. And what's great about the National Pig Centre is we have a conference suite. It can hold, I think, up to 80 to 100 at a push people. So we do have space to host events, and we will be looking to do many of those now in the months to come, now that we're sort of over the pandemic and any restrictions that were formally in place. So do watch the space, and we will continue to update our audiences via Twitter on what's coming up. The next question, so we have, it looks like somebody who's worked with pigs in their early life. So thank you for the webinar on the NPC, Katie, from this particular participant. I've worked in pigs for the first years of my life. However, I studied theology and religious studies at the University of Leeds. My question is, you mentioned zinc oxide supplements and its replacement. Do you also give little pigs iron supplements, and if so, how? Is it a dietary supplement or an injection?
Yep, we do. So the piglets will receive an iron injection within the first few days of life. And the reason for that is because they're being reared indoors. They don't have access to the iron in the soil. So all of our indoor pigs will receive an iron injection. Amy, that I mentioned, I don't know if she's here, but is interested in looking at whether the outdoor pigs would benefit from an iron injection as well. So we'll be looking at the differences between indoor and outdoor, hopefully somewhere down the line. But yes, so they do get an iron injection within the first 72 hours of life, and that is via injection.
If we gave our outdoor pigs an injection, would they just grow astronomically big really quickly. We'd have some strong pigs. Lovely. The next question is, do you know what the increase in piglet mortality is on the outdoor farrowing system without crates?
Yeah, it's a lot harder to know exactly outdoors, because the sows are giving birth within the ark. We don't have as easy access to get in and see what the mortality is. It will be higher than indoors. We're somewhere between 10 to 12% mortality indoors. It will be higher outside. I couldn't give you the exact number off the top of my head, but you've also got issues with the weather, with any predators. There's also a whole range of other things outside that increase mortality as well. So litter size does tend to be smaller outside at weaning.
Lovely, thank you. And I've just spotted a follow-up message from Ruth, who works with us on the wider farm, about the open days. So we will be having some open days looking at the regenerative agriculture that we have on the arable side of the farm. So it's worth mentioning that the National Pig Centre is part of a mixed farming system, so plenty of opportunity to come and visit us through Ruth's project. And I know that Sarah has posted a link to our 3D version of the tour, which is available online in 2D and 3D actually. So please do click the link and take a look. It looks like that is all of the questions. I'll give it a moment if anybody does have any last minute thoughts that they want to post into the chat session. For now, I don't suppose there's any closing comments from you, Katie, or any take-home messages that you want our audience to go away with today?
Oh no, I just hope you've enjoyed lots of pig pictures. So I always enjoy to get them in there. But no, just really wanted to share with you the work that we're doing, the emphasis on the sustainability of the pork production that we're going for. And yes, to thank you all for your engagement.
We've had one more question just in the nick of time. Could I just quickly ask, what do you do with our pig slurry, and is there potential to do any anaerobic digestion on site?
So the pig slurry at the moment is spread on the land, and it's in an exchange for straw for the sows. Is there potential to do anaerobic digestion? I'm not entirely sure on what we'd need legal wise, but yes, there is potential to do it. I don't know, Stefan, if you want to jump in on that.
Yeah, absolutely. There's quite a lot of work going on. We can what we can do with the slurry, and anaerobic digestion is certainly one of the things that can happen. Also in conjunction with the N2 applied unit that you mentioned. So yeah, it's certainly something that we're looking at.
Thank you, Stefan, for jumping in there. There's another little flurry of questions, so I'll quickly rattle through them. We've got a couple of minutes left. There are lots of cultural connections between pigs and humans, also cultural enrichment or toys for the pigs. Now, is that a potential area of research?
Yes, it is. So we did some work last summer putting chew toys in for the pregnant sows. So half of them had access to these chew toys, the other half didn't, and we looked to see the impact on marks on the body, so for aggression with the pigs. We're always putting different types of enrichment in through all the different life stages as well to keep the pigs interested. I'm also really keen to get some scratching brushes out there. So you see them a lot with dairy cows, but really keen to get some brushes in there, particularly for the pregnant pigs who like to have a good scratch, either from us when we're in there or from the walls. So yep, enrichment is a big thing that we're looking into.
Thank you very much. Sadly, we've run out of time now, and there's a lot of love for you in the chat there, Katie, so please do take a look at that. And we'll draw the seminar to a close now, and we will pick up any questions that we've missed after the session and respond directly. But before we close, I just want to give a special thanks to Katie again for such a brilliant presentation, and to GFEI for hosting the session today, and to all of you for attending and participating. It's been a really good seminar, and please do feel free to reach out to any one of us for any follow-on queries. And in the meantime, I hope that you enjoy the rest of your day. Thank you very much, and goodbye.
Thank you, Susanna.
Thank you.
Thanks. Bye.